Hello, I'm Martin Blaser. I'm the director of the Center for Advanced Biotechnology and Medicine, or CABM. Today, I have the pleasure of speaking with CABM resident faculty member, Dr. Hiroko Nobuta. She is an assistant professor in the Department of Neuroscience and Cell Biology at Robert Wood Johnson Medical School. Hi, Hiroko.
Hi, Marty. Thanks for having me.
Yeah, great. So I just want to start. Maybe you can tell everybody about your early education and what you were doing before you came to CABM.
Yeah, of course. I grew up in Japan, where I finished local high school. Then I attended a city college in Los Angeles, California. It was called Santa Monica College. Then there, I finished my basic classes and then transferred to California State University in Long Beach, California. So I majored in psychology there because I was interested in human cognition, but then I realized that the cognition really occurs at the level of brain cells. So I majored in neuroscience in graduate school. And I finished my PhD at University of California, Los Angeles, where I met one of the original glia cell biologists, John DeVilliers, who taught me the diversity of brain cells and then how glia cells, which were thought to be only the support cells for the neurons at the time, could cause awful neurological disorders. So my dissertation was on like a glia cell communication during inflammatory insults to the brain. Then I continued working on neuroscience questions as a postdoc. That was at University of California, San Francisco. There, I had a very productive collaboration with the Stanford University. I eventually became a shared postdoc between UC San Francisco and Stanford. So also there, my interest remained in glia cells of the brain. And then I combined with human iPS cell technologies and I studied neurological disorders affecting glia cells.
Great. And so how long have you been at CABM and what brought you here?
Yeah, so I came to CABIN in September 2020, so I'm kind of new. So from my first visit to CABIN, the members of the CABIN, not only the faculty, but also the support team were very, very welcoming. And then they were enthusiastic about helping me succeed my research project. Then I later found out that I was one of the 4 new faculty recruited in 2020. And then all of us happened to be parents with small kids. So I made connections with them quickly. And then I thought the CABIN was a great match and a great environment.
Great. So now that you're here, what's the main focus of your lab?
So my lab focuses on developing new therapies for brain disorders, especially the ones involving the specialized glia cells called oligodendrocytes. So oligodendrocytes are a type of glia cells that ensures smooth communication among neurons. But then the disorders like multiple sclerosis, leukodystrophies, and white matter injury associated with preterm birth, they all damage oligodendrocytes. So I would like to know how we can restore the function of oligodendrocytes. And not only that, very little is known about how oligodendrocytes develop in the human brain. So I am studying very fundamental questions about how oligodendrocytes mature during human brain development.
Yeah, great. So those are definitely important questions. So how are you approaching it experimentally?
Yeah, so I'm taking a new approach to study oligodendrocyte disorders because oligodendrocytes are in the brain, and so we cannot access them directly. So we made a new method to convert skin cells into oligodendrocytes in a dish. So when we use this conversion method, the skin cells resemble oligodendrocytes in many characteristics, and then we can even mimic oligodendrocyte disorders if we use skin cells from the patients with disorders. So I think that this is a new tool to study inaccessible cells and then their behavior in the lab.
Great, terrific. So using this new approach, what new project are you particularly excited about?
So I'm excited to use these engineered oligodendrocytes to find drugs that might help patients. So we commonly use animal models to study diseases and search for drugs, but then they often fail once they go to clinical trials, maybe because the animal models are different from human disorders. So by using the oligodendrocytes from patients themselves, I'm hoping to find candidate drugs that might actually help treat them.
Yeah, great. So let me just ask you, for possible future students or trainees. How do you propose to interact with students and trainees in your lab?
Yeah, so the thing is, at the time I started in September 2020, it was in the middle of COVID-19 pandemic. So I had to tweak how I interact with students who were very interested in doing research but could not come into the lab. So I decided to have online meetings where we review research literature and discuss the data we have collected so far, and then discuss the plans, what we can do remotely, and then also when we can finally come back to the lab. But I think so overall, the members of the lab are connected still, mostly virtually, but they show enthusiasm in the lab work. So I'm hoping I can welcome them back to the lab soon.
Yes, and when people come back to the lab, how will what will those interactions be like?
Oh my God, my lab is a wet lab, meaning a lot of bench work, rather than like data mining or computer based research. So we're gonna be in the lab working on the bench almost like 7 days a week. So it's gonna be a very not gonna be a quiet lab.
Sounds great. So now one last question, which is a little weird. I apologize in advance, but you're still a very young person. But imagine it's the end of your career, and we have to write
Yeah.
your scientific obituary. How would you like to be remembered for your career?
Yeah, as a new independent researcher, I have still a lot to learn, especially to be a good mentor. And that's one of the things I would like to be remembered as a mentor who helped make good scientists for the future. But I also would like to be remembered for the contribution in therapeutic development in oligodendrocyte disorders, because many oligodendrocytes do not have effective treatments and the patients really suffer from neurological symptoms. So if I could contribute to their health, I'd be very happy.
Yeah, great. Terrific. Well, that really concludes this session. So thanks so much, Hiroko.
Oh my God, thank you so much for having me.